Hello friends, welcome to my channel ChemStore. Hope you have followed my previous video on bioinorganic chemistry that is regarding with essential elements. Today let us see the next part that is about respiratory pigments. Here we have the overview of today's presentation. First, we will discuss about the need of respiratory pigments or oxygen uptake proteins in various biological systems. Then we will discuss about the definition of respiratory pigments, then structural and function aspects of various respiratory pigments, namely hemoglobin, myoglobin, heme and hemocyanin. Then we will go to the next topic, that is oxygen binding curves of hemoglobin and myoglobin. Then we will see the structure function relationship regarding the function of these respiratory pigments, how the structure varies or how the structure will be in biological systems. So at the outset, let's see what are respiratory pigments or oxygen uptake proteins. These are the proteins present in certain vertebrates and invertebrates to transport oxygen from respiratory organs to tissues. And then next question rises, why do we need respiratory pigments? What is the need of these respiratory pigments in biological systems? Oxygen cannot be carried inside the body to meet the metabolic demand. Moreover, oxygen cannot diffuse in our body as our skin is very thick. Hence, some channel must be there to carry it. What are the properties of a good oxygen carrier? So, it should bind oxygen at high partial pressure of oxygen. Then it should not oxidize cellular components, means it should not react with them. Finally, it should give up oxygen wherever it is required inside the body. That means a good oxygen carrier gives up oxygen on demand. Then what are these respiratory pigments structurally? Usually these respiratory pigments contain two parts. One is prosthetic group and the second one is protein chain. Prosthetic group is also called as metalloporphyrin, in which porphyrin ring chelated to a metal ion. The porphyrin ring consists of a macrocyclic pyrrole system with conjugated double bonds and various groups attached to the perimeter. This porphyrin ring with metal ion or prosthetic group is attached to a protein chain. In certain respiratory pigments like hemerythrin or hemocyanin, there may not be prosthetic group. The metal atom is directly bound to nitrogen of amino acids. So the next, let us see various respiratory pigments present in biological systems. So here we have four different respiratory pigments, namely myoglobin, hemoglobin, hemerythrin and hemocyanin. So hemoglobin and myoglobin are present in several vertebrates, whereas hemocyanin is present in certain mollusks and arthropods, whereas hemerythrin is also present in some of marine invertebrates. Now let us compare and differentiate the properties of these oxygen carriers. So different properties are there, like metal atom present in the respiratory pigments, then metal oxygen ratio, like that. So first, what is the metal ion present in different respiratory pigments? In myoglobin, hemoglobin and hemerythrin, the central metal atom is iron. Then in hemocyanin, it is copper. Then coming to the next property, that is metal oxygen ratio. In myoglobin, one oxygen molecule is bound to one iron atom. That means the ratio is 1:1. That means Fe is O2. Whereas in hemoglobin also, one iron atom is bound to one oxygen molecule. That is 1:1 ratio again. But in hemoglobin there are 4 heme units. That means 4 iron atoms are there in one hemoglobin molecule. So the ratio will be 1:1, but 4 oxygen molecules for 4 iron atoms. So it can be written as FeO2 taken 4 times. Then coming to hemerythrin, one oxygen molecule requires 2 iron atoms to be bound. So the ratio can be written as 2Fe is to O2. In hemocyanin also, 2 copper atoms are required to bound one oxygen molecule. So that is about metal oxygen ratio. Then coming to the next property, that is oxygen state of metal. So in hemoglobin, myoglobin and hemerythrin, iron is in +2 state, that is iron 2. Whereas in hemocyanin, copper is in +1 oxidation state. That means all these metal atoms are in lower oxidation states in respiratory pigments. Then ligand unit. Myoglobin and hemoglobin have porphyrin rings as that is chelated units as ligands, but in hemerythrin and hemocyanin, though their names contain heme, there is no prosthetic group or heme present in the respiratory pigments. The metal atom is directly attached to the nitrogen of amino acid. So we can write it as just protein side chains. The number of units present in each respiratory pigment. In myoglobin, there is only one subunit is there, that means one heme is there, whereas in hemoglobin it has 4 subunits, that means 4 heme units are there. So in hemerythrin, the number of subunits is 8, whereas in hemocyanin it is variable, that means it varies from one animal to another. The next property is molecular weight. So molecular weight of myoglobin is around 17,000 AMU, whereas coming to hemoglobin, as it has 4 subunits, it may be around 4 times of 17,000, that is approximately 65,000. Then hemerythrin, it has 8 units, that's why its molecular weight is 8 times, that means 1,08,000. Whereas hemocyanin, the molecular weight is also variable from 4 lakhs to 90 lakhs. So the last one is color. So the color is varied from when the respiratory pigment is oxidized, it exhibits one color, and when it is in deoxy form, it is in some other color. So in myoglobin and hemoglobin, when they are oxidized, they are red in color, whereas in deoxy form, they are in red-blue color. And hemerythrin, when it is in oxy form, it is in violet-pink color, and it is colorless in deoxy form. And coming to the last one, that is hemocyanin, it is blue in oxy form. So we can see the blood of some certain arthropods like cockroach, etc. The color is blue, and we can find that sometimes it is colorless. Well, that means the blood is deoxy form, in deoxy form. So let us see in detail about structure and functions of various respiratory pigments. First, we shall discuss about the structure and functions of myoglobin. So myoglobin is present in vertebrate vessels, and its molecular weight is 17,000 AMU. And the protein chain containing 153 amino acid residues are folded about the single heme group. The prosthetic group present in myoglobin is in heme, which is an iron 2 complex of protoporphyrin 9 ligand. So central metal atom is iron, and the ligand is protoporphyrin 9 ligand. The tertiary structure of a protein consists of 8 alpha helical regions and 6 short non-helical segments. So here we can see the structure of heme. So in the first diagram, it is the chelated ligand only, in which protoporphyrin 9 is present. 4 pyrrole rings are there connected with methylene groups, and several groups are there attached to the perimeter. So it is without central metal atom, so no iron ion is there. Then in the next picture, we can see the respiratory complex, that is respiratory pigment along with iron atom, so in which 2 hydrogens are not there. So it is deprotonated protoporphyrin attached to iron. So this is generally we call it as heme B. Some other heme molecules are there, but it is quite common in several biological systems. So here let us discuss about the coordination of iron with several nitrogens and some other groups. So there are 4 nitrogens. Iron has 6 coordination sites, among which 4 coordination sites are occupied by the 4 nitrogens of pyrrole groups. And the fifth nitrogen is a fifth coordination site is attached to the nitrogen of imidazole of histidine that comes from the protein chain. Then sixth coordination site is vacant. So it is actually in this picture it is already shown that it is bound to oxygen. So the sixth position is for oxygen molecule. So here we can see along with this protein chain in which heme unit is surrounded by this protein chain. So again we can discuss the structure of hemoglobin. That is formation of the complex inverse deproteination of 2 in NH groups, what we have already seen in the picture. Then at the center of the heme there is an iron atom. Then 4 of the 6 coordination sites around this atom are occupied by nitrogen atoms from a planar porphyrin ring. So it has a porphyrin has planar structure. Then the fifth coordination site is occupied by a nitrogen atom of imidazole from a histidine side chain. Then the last coordination site is available to bind an oxygen molecule. And then let us see the functions of myoglobin. So as we know, transport of oxygen is the main function of hemoglobin. Apart from this transport of oxygen, it has several other functions also. So the first one is storage of oxygen. So myoglobin present in muscle tissue is responsible for the storage of oxygen. So it stores oxygen and releases whenever it is required. Then the second and the foremost proper function is transport of oxygen. So it transports oxygen across the cell membranes. So that means myoglobin acts as an intermediate between blood oxygen transport protein, that is hemoglobin. So when hemoglobin brings oxygen from lungs to tissues, this myoglobin carries that is oxygen from tissues to the cells. So it is responsible for the tissue oxidative phosphorylation also. Then the next function is buffering capacity. That is, it buffers the partial pressure of oxygen within the cell. Now let us see the structure and functions of hemoglobin, the next respiratory pigment. So here we can see the structure of hemoglobin, where primary, secondary and tertiary structures are as in the case of myoglobin. But it is a tetramer of myoglobin. That means hemoglobin has 4 subunits. So 4 myoglobin units are there. Let us see the structure in detail. So myoglobin consists of 4 protein chains, each about the size of myoglobin molecule, and its molecular weight is about 65,000 units. And it is a tetramer of myoglobin, as we discussed earlier. Then it has 4 subunits, among which 2 subunits are designated as alpha, and which they have approximately 141 amino acid residues, and 2 beta units approximately having 146 residues. Just let us see the picture once again. So here these two are beta globins, in which beta chains in which only 146 amino acids are there. Then coming to the next two chains, those are in orange, those are orange in color. These are designated as alpha, in which only 141 amino acids are there. So it is a tetramer of myoglobin. Then each of these subunits can bind to one molecule of oxygen. That means 4 iron atoms are there to bind 4 oxygen molecules. So let us see functions of hemoglobin. So as usual, transport of oxygen from respiratory organs to the tissues. So in human beings, hemoglobin carries oxygen from lungs to tissues, and it hands over to uh, myoglobin. So it acts as carrier of oxygen from the organs of respiration to the tissues, where oxygen is used in various metabolic reactions. Then the second one is it has another function which myoglobin does not have, that is transport of carbon dioxide back to lungs. But this function is mostly done by globins but not by the heme. So that means protein chain carries carbon dioxide back to lungs. Then buffering capacity. So it is hemoglobin is an effective buffer. It maintains the pH of the blood at 6.8. So it maintains its isoelectronic point is 6.8. So it keeps the pH as 6.8. And finally, it has another function, that is viscosity of blood. So it maintains viscosity of blood for proper circulation in veins and arteries. The next important concept is about oxygen binding curves of hemoglobin and myoglobin. So the circumstances under which hemoglobin and myoglobin are required to bind and release oxygen are very different. That means hemoglobin carries oxygen from lungs to tissues, whereas myoglobin carries oxygen from tissues to cells. So hence these two iron proteins have different binding constants as a function of oxygen partial pressure. In the lungs, partial pressure of oxygen is high, but in tissues it is very low. So oxygen is carried by hemoglobin from lungs to tissues where myoglobin is present. So hemoglobin is a tetramer of myoglobin, hence the oxygenation reaction can be expressed as, so in lungs where hemoglobin binds to oxygen molecules, so it can be the equation can be written as Hb plus 4O2 giving rise to HbO2 taken 4 times. So here Hb means it has 4 Mb units, that is myoglobin units. So we can write it as HbO2 taken 4 times. So in tissues it can be written as HbO2 taken 4 times plus 4 Mb, means 4 myoglobin molecules interact with oxygenated hemoglobin to give 4 MbO2, that means 4 oxygenated myoglobins where oxygen is free from hemoglobin is free from oxygen. So what is the secret behind these reactions or transport of oxygen of myoglobin and hemoglobin? Here affinity of myoglobin for oxygen is very high. So it must have a greater affinity for oxygen than hemoglobin in order to affect the transfer of oxygen at the cell, where the oxygen concentration is very low or partial pressure of oxygen is very less. So the equilibrium constant for the MbO2 complexation can be given as Kmb is equal to MbO2, that is molar concentration of oxidized myoglobin by molar concentrations of myoglobin, the product of myoglobin and molar concentration of oxygen. So Kmb is equal to MbO2 by MbO2. As we all know that square brackets indicates molar concentrations. So here we have the oxygen binding curves, whereas this red line indicates the oxygen binding curve of myoglobin, whereas this blue one indicates the binding curve of oxygen binding curve of hemoglobin. If the total amount of myoglobin, that is Mb plus MbO2, is held constant, while the concentration of oxygen is varied in terms of partial pressure, so the curve is obtained as shown in the figure. So here we can see different curves here. So the oxygen binding curve of myoglobin is hyperbolic, whereas it is sigmoid. It is in S shape for hemoglobin. So here we can write the equilibrium constants of both hemoglobin and myoglobin. So myoglobin and hemoglobin have slightly different properties due to their different structures. So these different properties are commonly described by the oxygen binding curve, and we can also call them as oxygen dissociation curve, because in this oxygen binding curve, from left to right it is oxygen binding curve, whereas from right to left it is oxygen dissociation curve. That means the respiratory pigment that is leaving oxygen at tissues or whatever it may be. Okay. So on this graph, the y-axis represents the fractional oxygen saturation of protein, while the x-axis describes the partial pressure of oxygen. So x-axis partial pressure of oxygen, whereas y-axis indicates the saturation of oxygen, saturation of these respiratory pigments. So this implies that myoglobin has high affinity for oxygen, which binds oxygen strongly and does not release oxygen very easily. So we can see the off saturation point, that is 50% saturated, 50% of myoglobin is saturated with oxygen at 2 point. So we can see this one. So this line, it is at almost at 2.8 millimeters of mercury, whereas for hemoglobin we can see that off saturation point is almost at 26. That means hemoglobin binds to oxygen at higher partial pressures, whereas myoglobin can easily bind to oxygen at lower concentrations or lower partial pressures of oxygen. So we can write an equation for this binding constant also. So K is equal to F. K is equal to F by 1 minus Fp, or F is equal to Kp by 1 plus Kp. So this equation is for hyperbolic curve for MbO2 binding. Myoglobin is largely converted into oxymyoglobin even at lower concentrations of oxygen. On the other hand, hemoglobin displays a sigmoidal curve. That means the curve is in S shape, and this curve means that hemoglobin has a lower affinity for oxygen when compared with myoglobin. This type of curve is a result of cooperative behavior of hemoglobin. So hemoglobin has 4 subunits, and there must be a cooperativity among these subunits. So that is meant by cooperativity. As each heme site is filled with oxygen, the other unoccupied heme sites of the hemoglobin become more likely to bind to oxygen. Conversely, as each occupied site on hemoglobin unloads the oxygen, the other occupied sites become more likely to unload. If one subunit starts unloading, the second unit will also get ready. So in this manner, the different heme groups on the same hemoglobin are said to interact or cooperate with one another to inducing, unloading or binding to oxygen. So myoglobin, since it consists of only one single heme group, it does not display any cooperative behavior. So this is preciously while hemoglobin is used as an oxygen carrier, while myoglobin is used as oxygen storage. So as it has no cooperative behavior, myoglobin is responsible for storage of oxygen. So the equilibrium constants of myoglobin and hemoglobin on oxidation complexation, we can write them as K Mb is equal to MbO2 by MbO, that is myoglobin into molar concentration of myoglobin into molar concentration of oxygen, as we discussed earlier. Whereas for hemoglobin, we can write it as KHb is equal to HbO2 taken 4 times by HbO2 to the power of 2.8. So here we have a different constant called 2.8, and this exponent, this 2 exponent is called as Hill constant. So actually we can write it as N, as the number of subunits is 4. For this particular respiratory pigment, we can write it as 2.8. So oxidation of hemoglobin, we write it as 2.8. Then what is this Hill constant? So this 2.8 exponent is called Hill constant, or we can say Hill coefficient. That is introduced by Archibald Hill in 1910 for the oxygenation of hemoglobin. The value, that is 2.8 for oxygen binding, results from the fact that a single hemoglobin molecule can accept four oxygen molecules, and the binding of the 4 is not independent. If they are independent, the equation can be similar to that is equation for myoglobin, that is as Kmb. As they are not independent, they are with cooperativity, that changes, that is binding of oxygen changes depending upon the side heme units. So if they act independently, they would give a curve identical to that of myoglobin, like hyperbolic. But it is the cooperativity of 4 heme groups that produces sigmoid curves. So at low concentrations, hemoglobin is less oxygenated. So as we have seen in sigmoid curves, and at higher concentrations, it is oxygenated almost to the same extent of myoglobin. So in these graphs, we can see here the two are so here the two are equally saturated. So that means at higher concentrations or higher partial pressures, both myoglobin and hemoglobin are having same affinity, but at lower concentrations or partial pressures, myoglobin has high affinity for oxygen, and hemoglobin has less affinity. Then let us move to the next point, that is effect of pH on oxygen binding. So this effect of pH is introduced by Bohr, that's why it is called Bohr effect. So the pH dependence shown by hemoglobin is known as Bohr effect in the honor of its discoverer Christian Bohr. So Christian Bohr proposed this effect of or influence of pH on oxygen binding. So we know very well about Niels Bohr, who was awarded Nobel Prize for his outstanding contributions in the field of quantum theory and atomic structure. This Niels Bohr was son of Christian Bohr. And more surprisingly, son of Niels Bohr, Auge Bohr, was also a physicist and received Nobel Prize in 1975. According to this effect, off saturation pressure increases with decreasing pH. That means when pH decreases, the off saturation pressure increases. If the oxygenation curve were pH independent, that means if that does not depend on pH, the efficiency of pigment as an oxygen transporter would have dropped to 50%. So the decrease in pH eases the release of oxygen. That means when pH is less, hemoglobin easily releases oxygen. When hemoglobin binds one proton for every two oxygen molecules released. This favors the conversion of carbon dioxide into the hydrogen carbonate. So carbon dioxide will be transported back in the form of bicarbonate or hydrogen carbonate. So it converts two protons, convert carbon dioxide into bicarbonate, so which will be transported back to lungs. So the Bohr effect can be studied in this through this graph. Affinity for oxygen decreasing with decreasing pH facilitating the release of oxygen in periphery or muscles. So here in this graph we can see the off saturation partial pressures oxygen at various pH values. For pH 7.6, that is which is red in color. So this one, pH 7.6, and its off saturation point is almost around 32, around 28 millimeters of mercury. Whereas for the second one, that is green curve, in which the pH is 7.4, the off saturation point is around I think it is 32, whereas for the last one, that is pH 7.2, that means the least one, that is in blue color. So there the off saturation point is at which the partial pressure of oxygen is almost 40 millimeters of mercury. That indicates the affinity for oxygen decreases with decreasing pH. So when pH decreases, the affinity also decreases. That means it easily gives up oxygen. So in lungs where the pH is 7.6, hemoglobin binds tightly oxygen, or it binds oxygen strongly, where in periphery where pH is very less, or that is 6.8 or something, it releases oxygen very easily. So then structure function relationship. So all these release or binding depends on the structure of hemoglobin, and this structure varies according to its function. So hemoglobin has the capacity to bind from 1 to 4 oxygen molecules. That means at a time it may not bind to 4 molecules, but it varies from 1 to 4, ranging from fully desaturated hemoglobin, that is deoxyhemoglobin, in which number of oxygen molecules is 0, and to fully saturated, that is fully oxygenated hemoglobin. So the dynamics of oxygen exchange is highly regulated by several metabolically derived factors that collectively define the oxygen demand of an individual's tissues. Among the key metabolic factors regulating the dynamics of hemoglobin's oxygen exchange reactions is oxygen itself. So the first motto is oxygen. When oxygen levels are high, the capacity of partially saturated hemoglobin molecule to bind oxygen disproportionately increases with the number of oxygen molecules it has already bound. In other words, when environmental oxygen levels are high, partially saturated hemoglobin molecules exhibit enhanced affinity for binding additional oxygen molecules. So, affinity for oxygen increases with higher concentrations of oxygen in environment. Then a specialized behavior referred to this behavior is called as cooperativity. That means structure itself changes according to the function. So just by adjustment of these heme units which are side by side. So let us see in detail. So we can name them as T state and R state of hemoglobin. So among the 4 heme units present in hemoglobin, 2 are bound to protein chains having 141 amino acids and labeled as alpha units, and the other 2 are bound to another protein chains with 146 amino acids which are labeled as beta units. So 2 alpha units and 2 beta units, but these units can be written as alpha beta units. That means one alpha unit and one beta unit will become a team. That is the 4 units can be divided into 2 alpha beta units. Upon oxygenation of hemoglobin, 2 of the heme units move approximately 100 picometers towards each other. That means these 2 units move closer uh, by 100 picometers, while the other 2 alpha beta units, they move apart or they separate by 700 picometers. So 2 units come closer by 100 picometers and the other 2 move apart or separate by 700 picometers. And these movements are the result of a change in the quaternary structure of the hemoglobin and are responsible for the cooperative effects. So the quaternary structure exhibited by the deoxy form is called the T state. That means maybe in tense state, and that of the oxy form is called R state. So R means relaxed state. So T means tense because it needs oxygen, that's why it is in tension. So we can call it as tense state, T state, and R, as it is having oxygen, it may get relaxed, that's why it is called relaxed state or state. So here we can see both the states. In the first one, that is here iron has no oxygen, it is without oxygen, whereas in the second form, that is R state, iron is bound to one oxygen molecule. So this is R state. And see, one more thing is that there is a distance between this iron molecule or porphyrin and the nitrogen or histidine. So there is a gap, whereas here there is no gap. That means nitrogen is almost attached to this the distance between nitrogen and iron will be less. Let us see in the next slides which we can explain in the name of Perutz mechanism. So Perutz has suggested a mechanism to account for the cooperativity of 4 heme groups in hemoglobin. So to understand the interaction between oxygen molecule and heme, and of course its influence on protein chain, we need this Perutz mechanism. So British biochemist Max Ferdinand Perutz has suggested a mechanism to account for the cooperativity of 4 heme groups in hemoglobin. So it is proposed on the idea that the interaction between oxygen molecules and a heme group can affect the position of the protein chain attached to it, which in turn affects the other protein chains through hydrogen bonds, etc., and eventually the tertiary and quaternary structure of the protein. So that means if one is bound to oxygen, it gets change in other heme units. So the key or trigger in the Perutz mechanism is the high spin iron 2 atom in an oxygen free heme. The radius of high spin Fe2 plus is too large to fit within the plane of 4 nitrogen atoms, as I said earlier. It is above the plane. Iron atom is above the plane. Then the iron atom is thus forced to sit above the center of heme group with a distance about 206 picometers between iron nitrogen of histidine. Furthermore, the heme group is doomed upwards towards the proximal histidine, as in the structure we can see in the next slide. The distance may be decreased to 198 picometers from 206 picometers. So here in this structure, so the distance between nitrogen and iron is 206, and the iron is in high spin state, and as its size is quite larger, this iron atom cannot be fit into. So this one is what's the porphyrin macrocycle, and iron cannot fit inside the porphyrin ring as it has larger size in high spin. Whereas in oxygenated state, in its OR state, it will be pulled inside the porphyrin ring, because then it may become to low spin state, and in which its size will be lesser when compared with high spin, and it gets inside the porphyrin ring. That means oxygen molecule pulls iron inside the porphyrin complex, and the distance between nitrogen and iron will be less, that is decreases from 206 to 198 picometers. So this is called Perutz mechanism, and this picture again we can see in another slide. So here in this picture we can see there is a distance between iron and nitrogen, that is histidine. So here we can see the gap, whereas in the second picture, that is in OR state, it is very closer to iron. Histidine is very closer. So here the distance is 206, whereas in the OR state it is 198 picometers. So in these pictures we can show both T state and OR state of hemoglobin. To understand very easily, so here is the picture that is maybe proposed by Perutz. So we can see this Perutz mechanism, we can compare this with a seesaw. So when it is in T state, that means the seesaw gets down, whereas the other side there is that hemoglobin in which there is no oxygen. If he moves for it may this seesaw may turn to above, and then this histidine will go will touch the ground, and their oxygen will be bound to hemoglobin, which becomes OR state. So it is just to understand how this oxygenated hemoglobin is in OR state, and deoxyform is in T state. Now then let us compare the structures and functions of hemoglobin and myoglobin. So in this picture you can see one heme unit surrounded by one protein chain which is having 153 amino acids, but in hemoglobin there are 4 heme units and surrounded by 2 different types of amino acids, 4 amino acids. So 2 green alpha chains which are having 141 amino acids, whereas 2 red beta units which are having 146 amino acids. So first let us see similarities, and then we will go differences. So myoglobin and hemoglobin subunits are structurally similar, That means the central metal atom is iron, and the macrocycle, porphyrin macrocycle is same, and they are having same functional groups in perimeters. So 8 alpha helix contain heme group. Myoglobin is monomeric protein, whereas let us see similarities first. Both hemoglobin, myoglobin are oxygen carrying respiratory pigments in vertebrates. In both the pigments, hemoglobin is present, heme is present, which is an iron 2 complex of protoporphyrin 9 ligand. Both the pigments are red colored in oxy form, while in deoxy form they are in blue red color. The function of both the pigments is to bind oxygen, but their physiological roles are different. Now let us see the differences. So there are structural differences as well as functional differences. First let us see structural differences. Myoglobin occurs as a monomeric protein, while hemoglobin occurs as a tetrameric uh, respiratory pigments or tetrameric protein. Myoglobin consists of a single polypeptide chain with 153 amino acids, and two types of polypeptide chains are there in hemoglobin. Two alpha chains with 141 amino acids, as well as two beta chains with 146 amino acids. And then functional differences. So hemoglobin transports oxygen in blood, while myoglobin that's oxygen transports oxygen from lungs to tissues, whereas myoglobin transports or stores oxygen in muscles. So myoglobin has additional responsibility of storage of oxygen. Unlike myoglobin, concentration of hemoglobin in red blood cells is very high. So hemoglobin is more than myoglobin in blood because it has to carry oxygen from lungs to tissues of all the organs. Myoglobin can bind one oxygen molecule, so called it is monomer, and while hemoglobin can bind four oxygen molecules, so called a tetramer. Myoglobin binds oxygen more tightly as it has higher affinity, and then hemoglobin has lower affinity. That's why it binds somewhat looser. So some more differences are there. Hemoglobin can bind and offload both oxygen and carbon dioxide. That means hemoglobin transports carbon dioxide back to lungs from organs. But myoglobin does not have this function. Myoglobin exhibits hyperbolic curve, whereas hemoglobin exhibits the sigmoid curve for oxygen binding curves, and hemoglobin exhibits Bohr effect, that is affinity changes with pH. But myoglobin does not exhibit Bohr effect as the pH in muscles and tissues does not change. So myoglobin has its function in tissues only, where the pH is 6.8 or around 7. So that's about hemoglobin and myoglobin, and let us move to the next one, that is hemerythrin. Hemerythrin, as we already discussed, that it is a non-heme iron protein present in some of the marine invertebrates. Hemerythrin has a molecular weight of 1,08,000 or 1,07,000 around, and it consists of 8 identical subunits and each containing 2 iron atoms. That means 2 iron atoms are required for carrying one oxygen molecule. So in the tissues it exists as monomer, dimer, trimer or tetramer. And so in this picture we can see this hemerythrin in deoxy form, and when it is oxygenated, so here is the oxygen molecule, it is bound to 2 iron atoms. So the next one is hemocyanin. This pigment is found in the lymph of most of the mollusks and arthropods, and it is not found. It is not confined to special cells, but exists as a freely dissolved solute. Because no need to have veins or arteries in arthropods or mollusks. It is directly present in the lymph. The molecular weight of these respiratory pigment is about, or it varies from 4 lakhs to, so that is 90 lakhs. So the central metal atom present in hemocyanin is copper, and 2 copper atoms are required to carry 1 oxygen molecule. So in this picture you can see 2 copper atoms binding 1 oxygen molecule. So finally, let us sum up, or let us recapitulate what we had discussed today. So we have discussed about the need of respiratory pigments, why we need respiratory pigments, then various respiratory pigments present in biological systems, structure and function of myoglobin and hemoglobin, and the comparative study also in detail, then oxygen binding house of both the pigments, Bohr effect and Hill coefficient, then structure and function relationship, that is T state and R state, and mechanism proposed by Perutz also discussed. So that is about respiratory pigments, and if you have any queries or doubts, you can call me at the same number, that is 9440798008, or you can simply drop me a message, or you can mail also pragathi.madur@gmail.com. So thank you once again. Thanks for hearing, and we will move to the next video, that is on photosynthesis. With that we can conclude bioinorganic chemistry. Okay, thank you.